So my title today is the role of embryo assays in ensuring media manufacturing quality. I'll also start by getting my affiliations and conflicts of interest out of the way. Like Giles said, I have many years of IVF and media manufacturing experience. I was, together with Dr. Avery, the first in the world to commercialize a serum-free and ready-to-use IVF media range for veterinary use. I am the CEO and CSO and co-founder of Arts Media Denmark. I worked 6 years at Origio Cooper Surgical when it was in Denmark. I established and was head of the bovine IVF research lab back then. I have board positions at the chairman of the Danish Society of Reproduction Fetal Development. I was on the board of IETS and AETE. Today, Arts Media was founded 4 years ago, majority owned by a range of fertility clinics doing 50,000 cycles annually. We have FDA approved and available at IVF Store, a one-step culture medium with a new SSR. We have our viscosity oil, sperm gradient, and in application process, semen wash, oocyte aspiration, maturation medium. So that's who I am and my affiliations. My aim of the talk is to highlight the use of embryo assays for uninvited events, obviously for media manufacturing, but embryo toxicity in the lab and in quality control procedures. This is a picture of bovine blastocysts on day 7, and you'll find that my heart lies with bovine embryo assay as the most valid one to test and evaluate performance of media, plastics, whatever you introduce into the lab. So the mouse embryo assay is required for any assisted reproductive technology device that you are releasing. So there are guidelines. We are monitored by FDA through notified bodies. We show in audits our books for quality control, and every single product has gone through numerous tests along with the mouse embryo assay as a requirement for every single release. The MEA test protocol, to summarize, is you obtain 1-cell or 2-cell embryos from sacrificed in vivo fertilized mice. A minimum of 21 embryos should be used in the assay. You include a control group using a medium for embryo culture during the ART procedures, following the same procedures as the test group. Culture embryos for 96 hours, 1-cell, or 72 hours for the 2-cell. And the MEA acceptance criteria is that more or equal to 80% of the embryos developed into expanded blastocysts. There is a guidance for industry and Food and Drug Administration staff on this as well. So the bovine embryo assay, a BEA test, I call it, versus the mouse embryo assay, the test method is that the BEA test uses the same method as is described in the FDA guidance for MEA testing, only with 3 major differences. The bovine oocytes are in vitro matured and in vitro fertilized versus in vivo matured fertilized zygotes from the mice. The second big difference is that the embryos are kept in the medium for 7 days or more in the bovine test. In the MEA test, it has a duration of 5 days if a 1-cell system is used. And third and last, the BEA test uses approximately 200 inseminated oocytes in 2 to 3 replicates, whereas the MEA test requirement is 21 fertilized mouse oocytes, zygotes per group, and just 1 replicate is required. So that is obviously the first huge or the biggest difference. And here is from slaughterhouse obtained immature bovine oocytes for a BEA assay. So the BEA test protocol is that you collect ovaries from slaughterhouse cows, and then in the laboratory, the oocytes are aspirated, all follicles larger than 3 millimeters. You get approximately 8 oocytes per ovary. They are subsequently matured, fertilized, and cultured. And post 7 days, culture, blastocyst rates, and kinetics and morphology are evaluated, as well as hatching rates. And I'll get into that system, how we do that later on. So the average blastocyst rate in the Bayer test is between 35 to 45%. It thus indicates a performance difference, whereas 80 or 90 or 100% blastocyst rate in the mouse embryo assay, it'll only indicate whether the medium is toxic or not. I'll give you an example later on. So the conclusion on pass, fall, or fail criteria is that in the Bayer test, the blastocyst rate must be equal to or higher than the control IVC medium. In the MIA test, the pass criteria is equal or above 80% blastocyst rate. So let me quickly run through the protocol. We collect the ovaries, we keep them in a plastic bag so there's no communication of the follicular fluid being diluted with the water. Then we do wash it in saline just before aspiration with a vacuum pump. We aspirate all follicles above 3 millimeters. Here you see the aspiration process. We search for the oocytes. After maturation, we have them looking like this. They are all expanded cumulus cells and they all stick together. And you can move 40 oocytes all together just in one pipetter of 60 microliter medium. To move on to the IVF and IVC, we have a non-capacitating semen wash. We simply spin down, we centrifuge twice and use that pellet for counting the semen solution of about 300 microliter we get from 2 semen straws. We fertilize with 2 million per milliliter, either in drops or in wells. And after an overnight fertilization, this is what the mature oocyte that in the previous photo were all sticking together. You see the semen has done the work and spliced all the cumulus cells. Denudation now occurs in the bovine, where it's in the human embryology. It's after aspiration of a mature oocyte. Here it's the zygotes that we denudate to get rid of the remaining cumulus cells from here prior to putting into culture. And to quote Dr. Avery, it's not a television show. We do not take a long time to discard oocytes, or we don't from the test. We also don't evaluate a lot. We are processing 200 presumptive zygotes, but you can get a quick indication of whether this is going to be a high or a low blastocyst rate, since we cannot, due to the dark content of the bovine oocytes, we can still get an idea of the fertilization rate by seeing this is almost a 2-cell. This is fertilized. You see a clear perivitellin space. Also here you see a polar body, and here you see a non-fertilized oocyte. It goes right out to the sonar. But to get a true picture and count, you need to do a stain and count the prune nuclei. Then we get 30 to 50% blastocyst rates after 7 to 8 days culture. And the result of the blastocyst, if they aren't used for Bayer, 50% will become pregnant if you transfer them. So there are huge differences in reproductive physiology between the mouse and a woman, and also between a cow and a woman. The most obvious similarity from a physiological point of view in a cow and a woman is the gestation time, and that they don't give birth to litters like mice. So should a batch variation occur, it'll not be detected in a MIA test, where release parameters is more than 80% blastocyst rate. In the Bayer test, however, a different as 35% versus 55% blastocyst rates indicate the two media are different. However, both media will have passed the MIA test. Hence, the MIA is a mere toxicological bioassay, whereas the Bayer also provides functional and performance data. So more and more labs are introducing ways to test their disposables and their media. It used to be they would perform MIA embryo assays. More labs are starting to also supplement with a bovine embryo assay. At the end of the day, it should not be necessary if the company, the provider of your art products, are strictly monitoring all the steps and the release parameters. It shouldn't happen that anything is released that is not fit for culturing embryos in. Comparison of embryo developed in human cow and mouse, there are huge differences as well. The oocyte diameter is more equal to cow and human. The start of the zygotic genome activation, time to reach a 2-cell stage, the blastocyst hours, hatching hours, and then implantation days is difficult to compare because there's a total different implantation and placenta formed in the cow. But the amino acid uptake and utilization differ among the species, and mouse embryos being very easy to culture with almost no nutrients at all compared to human and bovine. Also, the mouse embryos are less sensitive to changes in pH. Both human and bovine are more sensitive, in particular fluctuations. And then the strains of mice and the developmental stage. The strains of mice used vary, and the stage of embryo used and the culture conditions employed may also affect the outcome. So obviously a test in the MIA where you use a 2-cell embryo is much less sensitive than if it's a 1-cell, or even if it's the bovine where in vitro maturation and fertilization takes place. This is a lot of data to comprehend. Just to give you an idea how we evaluate in the BIA test, we combine kinetics, morphology, and the blastocyst score provides a much better impression of the effectiveness than looking at the blastocyst rate alone. So this system was developed by Dr. Avery and Professor Greve back in 2004 at Copenhagen University. These are the number of oocytes per week that is done. So these are 2 replicates. These are different kinds of media used. There are 4 groups. And then from these oocytes, we count the blastocysts, mark the non-blastocysts for our statistical analysis. And then we for the kinetics, we score 3, a non, expanded, and a hatch, and we grade a poor, a good, and an excellent. Then we put them into the calculation, and this is we get for the same media. So each group of media have about 200 oocytes in each group. The blastocyst rates are 39, 37, 28, or 35. So these 3 groups all are more or less identical. They are not statistically different. However, commercial medium 2, which is a medium that has passed the MIA test, which is sold to human clinics, I would definitely prefer not to have. I would choose one of these instead. The other interesting feature when you add kinetics and morphology score and the arbitrary score is that though this blastocyst rate is lower than the commercial medium, it still has a higher arbitrary score, and you can see it's due to higher kinetics and a better morphology. Not so much better morphology, but in particular a higher kinetics. And then that being said, higher kinetics is not necessarily good, but there are more parameters to evaluate for your media, for your plastics, for your pipette, whatever disposables you're testing, than if you just have plus 80% blastocyst rate. So reliability and consistency. The final performance is above all demonstrated in the bovine embryo assay test. So in media manufacturing, final control steps are crucial for quality assurance, and standard tests like pH, endotoxins, fungal, and sterility are routine. So it should not happen that you have a media that fungal or bacteria arises from, because there are numerous tests performed before they are released. And again, the MIA test would catch many of these. Then assessing performance is equally important, and I think it's often overlooked, and therefore I really welcome that more and more are looking towards the BIA test. I think I touched upon how I think MIA lacks the sensitivity And again, the huge numbers of different 21 oocytes compared to 900 or more. And then you evaluate both performance and toxicity. So ensuring laboratory safety and quality assurance in media verified by embryo assay testing, it should not be necessary in your clinic that you also have to test your products. You should be able to rely on your supplier. I understand the wish to be in additional control of this. I have seen a tendency that you are looking to the website for the media supplier. When is the most fresh medium? When is the next manufacturing going to happen? And you see, I want from that batch. Some media have 18 months of shelf life, some have 24. This is not a bad thing. If that medium in the last month of its shelf life is not performing equally to its first month, then that company overall has a problem, because it's audited by notified bodies on behalf of medical agencies and FDA, and their tests should prove that the quality and the performance and the tox and all the sterility is maintained. Otherwise, that shelf life could not have been determined correctly. So that is the shelf life. Preservation with glass packaging ensures superior preservation of culture media. There's less VOC, and plastic nanoparticles is a huge thing now being found everywhere. So this is another way you can invite uninvited things into your lab. I put this just recently in my slide, having attended a human conference. Media that's been aliquoted for 1 hour to a plastic vial will give a significant decline in blastocyst rates in the beer test. So do not aliquot. Consider these SOPs if you have SOPs leftovers from when there were bad oil batches, or you had to wash the oil, or is it just a leftover? So consider changing this and get media from glass. But this is this was a very interesting finding that we did. It showed in the bovine test. And then to change from using the media with the shortest shelf life, you will avoid inviting trouble into your lab if you use a longer shelf life, because there are good witnesses programs, but if these programs could also, if you could talk to each other among the clinics, there is a lot of security that you have a batch that is used in labs worldwide. So if you have a medium that has a stability and a shelf life of 18 months, think of how many other colleagues are using this. And when you have uninvited problems in your lab, worst problem is that you have seroblastocyst or embryo development. The first thing you say is, what's wrong with the oil? The next thing you say is, what's wrong with the medium? And then you start looking to other things. So imagine you could rule out that, because you know you are using a batch that is used in hundreds of other labs worldwide. So the freshness of media should remain consistent between the newest and older media batches within the shelf life. So I hope also with this uninvited that you have a very poor embryo development, that I can invite along with the witness systems, maybe that you can talk to each other and find the assurance in the media you use is being used by many colleagues around the world for many, many weeks. So on behalf of IVF Store and Arts Media Denmark, thank you for your attention, and thank you again for the moderators and for i3 for the opportunity.